There is something living inside roughly one-third of all humans alive right now. It does not cause fever. It does not cause pain. It produces no rash, no cough, no swelling you could point to and say, there. That is the problem. Most people who carry it will never know it is there. Their doctors will never test for it. Their entire lives will pass without a single symptom they could name. What it does is quieter than that, and far more disturbing. It changes who you are. Not in a dramatic way, not in a way you would notice from the inside. It does not take over your body like a fever dream. It does not whisper instructions into your sleep. What it does is subtler, and in many ways that makes it worse. It adjusts small things. The edge of your fear response. The weight of a risk before you take it. The particular chemical texture of a decision that feels, from the inside, entirely your own. To understand what this organism actually does and why it does it, you have to start somewhere unexpected. You have to start with a rat and a smell. The organism is called Toxoplasma gondii. It is a single-celled parasite, microscopic, unremarkable in appearance, and it has a problem. To sexually reproduce, to complete its life cycle, it needs to be inside the intestine of a cat. That is the only place on Earth where it can do what it needs to do. Every other host is a biological dead end. A rat, a bird, a human. These are detours, temporary vessels. The parasite needs to get back to the cat, which means it needs the cat to eat its current host. So it does something that should not be possible for an organism with no brain, no nervous system, and no awareness of its own existence. It solves the problem. A healthy rat has a hardwired, deeply ancient fear of cat urine. This fear does not require experience. A rat raised in a laboratory that has never encountered a cat in its life will freeze and flee from the smell of cat urine on its first exposure. This is not learned. It is written into the rat at a level below thought. Toxoplasma rewrites it. Inside a toxoplasma-infected rat, something changes in the amygdala, the brain's threat detection center. The fear response to cat odor is not merely suppressed. It is converted, inverted. What was once a survival alarm becomes something closer to attraction. The infected rat does not avoid the cat, it moves toward it. The cat eats the rat. The parasite reaches the intestine it needed. The cycle completes. And Toxoplasma gondii has just solved a complex navigational problem using the brain of another species as its tool. That is what it does to rats. Now here is the question no one wanted to ask. What does it do to us? In the early 1990s, a Czech evolutionary biologist named Jaroslav Flegr started noticing something wrong with himself. Flegr worked at Charles University in Prague. He had tested positive for toxoplasma, a common enough finding given that roughly 30 to 40% of the Czech population carries it. Most physicians treat a positive result as a non-event in healthy adults. The acute phase passes. The parasite enters a latent stage. Life continues. But Flegr began to suspect that latent did not mean inactive. He noticed in himself what he described as a diminished response to threatening situations. A blunting. Not fearlessness exactly, more like a recalibration of what registered as genuinely dangerous. He was, by his own account, crossing busy streets without the caution he knew the situation warranted, acting in small but measurable ways differently than he once had. He was a scientist, so he did what scientists do. He tested it. What Flegr found across years of published, peer-reviewed research, studies involving thousands of subjects, comparing toxoplasma-positive individuals against uninfected controls, is difficult to dismiss. It is also difficult to fully explain. Infected men in his studies showed a measurable shift in personality profile, decreased novelty-seeking, increased suspicion, a specific blunting of risk assessment that did not feel from the inside like recklessness, but produced reckless outcomes. Infected women showed something different, something almost opposite, increased warmth, greater social openness, a personality shift in the direction of what standardized testing calls agreeableness. The same parasite, two different behavioral signatures, depending on the hormonal environment it finds itself in. And then the traffic data. In a 2002 study published in BMC Infectious Diseases, Flegr and his colleagues compared 146 toxoplasma-positive subjects against 446 uninfected controls, all Czech military conscripts, a relatively controlled population. The infected group was 2.5 times more likely to have been involved in a traffic accident, not slightly more likely, 2.5 times. When you read that number, something in your reasoning will immediately reach for an alternative explanation. Sample size, confounding variables, coincidence. That is a reasonable instinct. Flegr himself would agree with the caution. These studies are largely correlational. They show a relationship between Toxoplasma infection and behavioral change. They do not prove, in the way a controlled experiment would prove, that the parasite is the cause. Running that controlled experiment, deliberately infecting humans with a parasite to observe behavioral outcomes, is not something that can be done ethically. So the correlation is what the science has. And the correlation is consistent. Replicated. Published in peer-reviewed journals across multiple countries, multiple populations, multiple decades. Flegr is not a fringe figure. He is a working scientist publishing in respected journals. The findings are not settled, but they are not nothing. They are something. Here is what makes them more than something. When researchers stopped looking at behavior and started looking at the brain, at what Toxoplasma is actually doing inside neural tissue, they found something that should not have been possible for an organism of its simplicity. Robert Sapolsky, neuroendocrinologist at Stanford, one of the most respected figures in behavioral biology, documented what Toxoplasma does inside the rodent amygdala in work he later described as one of the most extraordinary things he had encountered in his career. The parasite does not simply take up residence in neural tissue, it does not simply form cysts and wait. It carries, encoded in its own genome, the instructions for producing an enzyme called tyrosine hydroxylase. Tyrosine hydroxylase is a key enzyme in the synthesis of dopamine. Let that settle for a moment. A single-celled parasite with no brain, no nervous system, and no evolutionary history inside a vertebrate body carries the genetic machinery to alter dopamine production in the brain it is living inside. This was confirmed in a 2009 paper published in PLOS ONE by Gaskell and colleagues, who identified and sequenced these tyrosine hydroxylase genes directly from the Toxoplasma genome. The genes are real. The enzyme is real. The dopamine alteration is real, at least in infected rodents, where it has been demonstrated with precision. In the rat's amygdala, this is what produces the attraction to cat odor. The fear signal is chemically replaced with something that activates reward circuitry. The rat does not stop being afraid because it becomes brave. It stops being afraid because its fear has been chemically converted into wanting. In humans, the dopamine picture is less precisely mapped. The cysts form in the amygdala and prefrontal cortex, the same regions, and the tyrosine hydroxylase genes are present in the organism. Whether they operate with the same mechanism in humans as in rodents is not yet definitively established. But researchers studying the Toxoplasma schizophrenia connection, E. Fuller-Torrey and Robert Yokin among them, who have published multiple studies across 2 decades showing elevated Toxoplasma antibody levels in schizophrenia patients compared to controls, have pointed to dopamine dysregulation as the most plausible mechanistic link. Schizophrenia is, among other things, a disorder of dopamine regulation. Toxoplasma may be quietly altering the same system. In most infected people, the effect falls far below the threshold of clinical disorder. It does not produce psychosis. It does not produce anything you could point to in a doctor's office and name. It produces something subtler, a slightly different fear threshold, a slightly different response to risk, a slightly different chemical weight on the decisions that feel, from the inside, like your own considered judgment. Now the twist. In 2006, a marine ecologist named Kevin Lafferty published a paper in the Proceedings of the Royal Society, B. Lafferty works at UC Santa Barbara and the U.S. Geological Survey. His expertise is in how parasites shape ecosystems, not human psychology. But what he did in that paper was take Flegr's individual-level behavioral data and ask a question that Flegr had not asked. If Toxoplasma infection rates vary dramatically across countries, and they do, ranging from around 11% in some parts of the United States to 45% in France to over 60% in Brazil. And if those infections produce measurable personality shifts at the individual level, then could infection rates help explain measurable personality differences between national populations? Lafferty ran the analysis. He found correlations between national Toxoplasma prevalence and aggregated personality scores, specifically in a trait cluster associated with neuroticism. Countries with higher infection rates showed higher average neuroticism scores. This paper has been criticized. The methodology carries real limitations. Correlations at the national level are vulnerable to confounding variables, and drawing conclusions about individuals from population-level statistics is a recognized statistical hazard called ecological fallacy. Lafferty himself frames the findings as a hypothesis, not a conclusion. Other researchers have noted that infection rates correlate with many other national variables: wealth, healthcare access, diet, climate, making causal attribution to Toxoplasma specifically very difficult. The paper is contested, but the question it opens cannot be closed again, because here is what it is actually asking. 1/3 of all humans alive right now carry a parasite that concentrates in the amygdala and prefrontal cortex, carries genes for dopamine synthesis, demonstrably alters fear response and risk behavior in rodents, correlates with behavioral differences and accident rates in human studies, and has been present in human populations since at least the dawn of agriculture. 10,000 years of cohabitation with the species that built civilization. How much of what we call human culture, the risk-takers who founded companies, the soldiers who charged, the explorers who sailed into unknown water, the decisions made in parliament and on battlefields and at the edges of cliffs, how much of that was shaped not purely by human minds, but by an organism optimizing for one thing and one thing only: getting back inside a cat? That is not a proven claim. It is not presented here as established science. It is a legitimate question raised by credentialed researchers, published in peer-reviewed journals, and not yet answered. That is somehow worse than if it were settled. There is one more data point. A 2010 study published in the Archives of General Psychiatry, led by Theodor Postolache and colleagues, analyzed data from more than 45,000 Danish women. Women with higher Toxoplasma antibody levels showed a statistically significant increased rate of suicide attempts. Not a trivial increase, a 54% higher rate compared to uninfected controls. The researchers were careful. They noted the correlational nature of the data. They pointed to confounding variables. They did not claim Toxoplasma causes suicide. What they said was, the association exists. It is real. It requires explanation. Dopamine dysregulation is one possible explanation. Altered risk perception is another. The exact mechanism is not established, but the pattern is there, quietly, in the data. Here is where you are now. You started this video as an observer. Someone watching a story about a parasite. Something out there. Something that happens to other people. To rats in laboratories, to statistics in Czech military studies, to numbers in Danish health registries. But 1/3 of all humans alive right now carry Toxoplasma gondii in their neural tissue. 1 in 3. That is the person next to you, or the person you spoke to this morning, or the face you see when you look in a mirror. You cannot feel it if it is there. You cannot locate it. No symptom announces it. The cysts are silent. The dopamine alteration is silent. The slight recalibration of your fear threshold, if it is happening, feels exactly like your own personality, your own judgment, your own considered, autonomous, freely chosen response to the world. And the part of you that would notice, the observer inside your own mind, the voice that watches your decisions and calls them yours, may itself be running on chemistry that was never entirely yours to begin with. There is something living inside roughly 1/3 of all humans alive right now. It does not cause fever. It does not cause pain. What it causes is something much harder to see, and much harder to dismiss. It causes you to feel like yourself,